So today I'm going to tell you about some research from our lab where we've been using mRNA metabolism as a way to do precision therapeutics specifically for haploinsufficiency disorders. Now, in my lab, we study mRNA, and mRNA is the mediator of genetic information between DNA and the cytoplasm. It conveys genetic information into the cytoplasm where it's translated by ribosomes to make proteins. And one of the most important aspects of mRNAs is that they go away. And the reason for this, and the reason why this is important biologically, is that if a signal in the nucleus it regulates the expression of an mRNA synthesis, then that signal is really irrelevant as long as the mRNA continues to exist in the cytoplasm and make protein. And so an integral part of gene expression is that an mRNA is timed to be destroyed. And that allows a linear flow of genetic information from DNA to protein. And my lab studies the process of how long mRNAs survive within the body as a way to understand gene expression and genetic control. And with all the information we know about how mRNA stability is regulated, we've used this information to go after therapeutic modalities that could impact various human disorders. And one place where this becomes very important is in the treatment of haploinsufficiencies. And a haploinsufficiency, of course, is where one of the two copies from either mom or dad's DNA has been mutated in such a way that either the mRNA is no longer made or the mRNA is made aberrantly, but the net effect is that you get no protein. And for about 300 different disorders, this is incredibly important for maintaining appropriate human health. And so we've gone after what we call mutation agnostic therapies, where what we try to do is increase the expression of the wild type copy of the mRNA through modulating its stability in order to achieve at least a twofold increase in the protein amount and therefore potentially amelioration of the haploinsufficiency. Now, a key component that controls how long an mRNA exists within the cell is a element that's present on the 3' or the end of every single mRNA, or at least most mRNAs in the human body. And this is called a polyadenosine tail or poly-A tail. And really all we need to remember for this talk is that if an mRNA has a poly-A tail, it's loaded on ribosomes and it makes a protein. If an mRNA loses its poly-A tail, then it no longer makes protein. And the poly-A tail is the key feature that tells the cell how long an mRNA should survive within the body. Once that mRNA goes out into the cytoplasm, that poly-A tail is being chewed by an enzyme that is depicted here by this Pac-Man that we call a deadenylase. And this rate at which it's destroyed is very specific to the given mRNA and again acts as a timing event. And so it keeps this mRNA expressed for, in this case, 30 minutes. And then as soon as that tail is lost, the mRNA is usually degraded or simply translationally repressed, but no matter what, it's not making a protein anymore. So how can we use that information in order to achieve a therapeutic modality? And so what we've done is we've essentially fooled the mRNA into thinking it still has a poly-A tail. And we do this by using a short antisense oligonucleotide that we simply polyadenylate. And so we add this poly-A tail to this oligo. And when the mRNA has lost its natural poly-A tail, this is still binding to the mRNA and mimics the function of the poly-A tail and keeps the mRNA translating as it normally would. And this would allow for an increase in the overall protein that comes from that wild type mRNA. And so we call this an mRNA booster or a poly-A tail mimetic, and it's a novel therapeutic candidate for the treatment of haploinsufficiencies. And the general idea is an antisense guide to a specific mRNA and a poly-A tail mimetic. And this is simply a poly-A stretch that we also can modify chemically in order to preserve the stability of this short oligonucleotide. This will then bind to a target gene, target mRNA, and enhance its overall translation. So in the first iterations of this type of experiment, we simply took RNA oligos that we would synthesize, and we used a enzyme called polymerase to add a poly A tail to these oligos. And you can see that can add a very long poly A tail, so without the poly A tail and then with the poly A tail. And if we introduce these um, alongside of reporter mRNA, so this is a reporter that just encodes a fluorescent reporter, and we're adding a poly-A-tail mimetic that's antisense to that reporter, and as well as a control. When we transfect that into cell culture, we can get a robust stimulation of that firefly luciferase mRNA in vitro, and this is in fact up to sixfold above the background. And so quite a significant stimulation of the translation of that mRNA by the poly-A-tail mimetic. Now, we want to use this technology not to look at reporter genes, but rather endogenous mRNAs, RNAs that are normally expressed within the cell. And so this is an mRNA that has no therapeutic value whatsoever, but it's an mRNA that we've studied a lot in my lab. We know a lot about it, and it's called LSM8. And so one of our first experiments was to develop a poly-A-tail mimetic directed against the LSM8 mRNA in cell culture. And you can see that we could develop tail mimetics that would stimulate the overall expression of the LSM8 protein. Now, we have then turned our attention to develop poly-A-tail mimetics against mRNAs that are associated with bona fide haploinsufficiencies, genes that are whose mutation manifests in a disease phenotype. And we chose to focus on MECP2, which has been implicated in Rett syndrome, CTNNB1, which is beta-catenin deficiency, pure alpha, and SYNGAP1 deficiency. And in each of these cases, we could make a poly-A-tail mimetic that would lead to a specific increase in the mRNA associated with the haploinsufficiency. And again, this was done in cell culture. Focusing a bit more on some of these specific targets, in the case of MECP2, we can get poly-A-tail mimetics that work in vitro, but also in vivo, in mice. And so this is an experiment where we did a tail injection of a poly-A-tail mimetic. And then in a wild type mouse, this is not a Rett syndrome mouse model, but it's a wild type mouse. And when you do a tail and vein injection like this, the oligonucleotide will go to the liver. And so if we harvest the liver and then perform a Western blot, you can see we can get a stimulation of the MHCP2 protein for this particular booster in the liver. In addition, we've done similar experiments with beta-catenin, and I'll talk more about beta-catenin toward the end of the talk, where we can develop in vitro poly-A-tail mimetics that will give us a dose-dependent effect on the expression of CTNNB1. And then in collaboration with Michelle Jacobs' lab at Tufts, we've injected those oligonucleotides into the tails of wild-type mouse and seen an increase in beta-catenin expression in the liver. In addition, we've looked at beta-catenin expression in cultured neurons and see that we can, with that same booster that I was showing for the wild-type mice, get a significant stimulation of the translation of beta-catenin in neurons. Beta-catenin is a transcription factor, and it regulates certain downstream targets. And not only do we see the increase in beta-catenin expression, we also will see the increase in its downstream targets, which makes sense because we are now increasing the expression of the transcription factor. In addition, we've focused a great deal of our attention on SYNGAP1 deficiency. SYNGAP1 deficiency, like many of these haploinsufficiencies, is a developmental encephalopathy that has very similar phenotypes to other disorders like Dravet and CDKL5, Rett syndrome, et cetera. SYNGAP is a key protein required for the appropriate function of neurons and allows them to communicate with each other. And many of the phenotypes of children afflicted with SYNGAP are similar to those other disorders I just mentioned, including autism-like behavior problems, intellectual deficiency, etc. One of the key points of these developmental encephalopathies is that they are often the result of sporadic germline mutations. And so they're not necessarily inherited disorders from mom or dad, but it's a sporadic mutation that happened in one of the two gametes. And this makes it a big challenge from a therapeutic standpoint because while these disorders are ultra rare, they often have a large number of mutations that can result in the particular indication. So, for example, there's well over several hundred different mutations that lead to SYNGAP deficiency, and there's only 17 to 1800 patients worldwide known to have SYNGAP deficiency. So, developing mutation agnostic approaches for the treatment of these haploinsufficiencies becomes a very attractive route to go. In addition, the genes like SYNGAP, as well as many other human uh, genes, have spliced isoforms. And so we need to be able to increase the expression of all of the various splice isoforms. And our technology uses poly-A tail mimetics that are binding to the 3' untranslated region, which is often shared among all of the isoforms. And so in theory, if we stimulate the translation of SYNGAP, we're in fact stimulating the translation of all um, four isoforms so that you can get appropriate function. Again, we were able to develop poly-A tail mimetics that would enhance SYNGAP expression. This is just an in vitro experiment in cultured neurons. And this particular poly-A tail mimetic gave a very nice, robust response to SYNGAP expression. We've also done target candidate screening to identify oligonucleotides within the 3' UTR that perform better than others. And this is a limited study because these experiments can actually become quite expensive. But you can see that we had tiled through the 3' untranslated region with these 5 different oligos, and this particular poly-A tail mimetic performs better than the ones that are around it. And then with SYNGAP, we've done experiments where we've directly injected the poly-A tail mimetics into the hippocampus of the brain using a brain perfusion approach where we have a nanotube that gets injected into the hippocampus, and that nanotube then leaves behind a superelastic permeable tube, which is then allows us to perfuse oligos into the hippocampus and then monitor the effects of the poly-A tail mimetic. And in these early experiments, um, sorry, this got duplicated for some reason, but in these early experiments where we just had 3 mice, you can see in the hippocampus the nice increase in expression of SYNGAP1 that it was also seen in other parts of the brain, but not seen in the cerebellum. We've repeated this experiment in many more mice, and just focusing on the hippocampus now, you can see that we can get a nice, at least twofold effect on SYNGAP expression in that region of the brain. Now, all of the experiments that I've shown you so far are using oligonucleotides that are fairly unstable, meaning that RNA in general is a very unstable molecule. It's quickly degraded by the human body. And so, in order to move toward a therapeutic modality, we need to have chemistry that would improve the overall stability of these molecules that we're trying to deliver. And so we've employed various modifications that are known to enhance the stability of mRNAs. This includes phosphorothioates and 2' methyl groups. And really, all you need to see here is that if we look at our first generation and second generation poly-A tail mimetics after a transfection experiment and then waiting a week, pretty much they're gone. But if we have these modifications that we've built into these oligonucleotides, they're very stable even after a week here. And we predict that they would be very stable in the body as well. And in the case of SYNGAP, these still function just as they do when they're unmodified. In addition, we wanted to verify that these modified nucleotides would still bind to the protein that's required for the function of the poly A tail, and that's a protein called poly A binding protein. And in this binding assay, you can see that um, the modified oligonucleotides bind poly A binding protein just as well, and if not better, than the unmodified. So if you're not familiar with dissociation constants, the lower the number it is, the better its binding. So this is in fact an increase in binding of that protein to the RNA. In addition, we wanted to see if we could shrink the size of these oligonucleotides. So poly A binding protein, this protein that we were studying over here that's responsible for poly A's effects, binds to every 25 A's. And so a poly A tail of 100 has 4 molecules of poly A binding protein on it. And what you can see though is that as we shrink the size of the poly A tail, going from 4 to 3 to 2 to 1 molecules of poly A binding protein, we don't take a big hit in the overall efficacy of the poly A tail mimetic, suggesting that we could get to a oligonucleotide, a fairly small size, would then allow us to develop it as a therapeutic candidate. In addition, these poly A tail mimetics have been put into these new forms of poly A tail mimetics have been put into mouse brain in very preliminary experiments, and we still see rescue of the SYNGAP phenotype in those mice. But we have to do a lot more data. We have to do a lot more optimization of these poly A tail mimetics to get above these rather subtle effects that we're seeing. Now, last but not least, returning to some of our recent, more unpublished work, both for SYNGAP, that I just showed you, as well as CTNNB1, we've used patient-derived neurons. And again, these are experiments we've done in collaboration with Michelle Jacobs. And in Michelle's lab, using these patient cells, she's seen that the modified versions of the beta-catenin poly A tail mimetics also stimulate the translation of that gene, advancing us toward possible developmental candidates. And we've done similar experiments as we did with SYNGAP, where we're trying to shrink the poly A tail size for beta-catenin, and we seem to be hitting a sweet spot somewhere between 25 A's and 50 A's in order to see a robust stimulation. But like SYNGAP, we also need to start working on these oligonucleotides to improve the overall efficacy and then lead toward a developmental candidate. And so what I've shown you then is poly A tail mimetics or mRNA boosters are a viable approach for continued development for the treatment of a variety of haploinsufficiency disorders. We've shown in mice that we can see significant stabilization and stimulation of the translation of the MECP2 mRNA, the beta-catenin mRNA, and the SYNGAP1 mRNA. And so we suggest that this technology represents a novel gene therapy that enhances protein expression. It's a first-in-class technology that leverages the power of RNA therapeutics. It's disease-modifying, mutation-agnostic, and a platform technology that is portable across numerous haploinsufficiency indications. And so with that, I'll just mention the people who did this work. Most of the work in this story was done by a postdoc, Bahare, shown right here, uh, with assistance from Sophie and Emil and Christian. And the people who have given us money to do this work are shown here. And I'll stop there and thank you for your attention.